Hello, students. A very good morning to all of you. So, today in this session, we are going to solve previous year questions on the chapter Human Health and Diseases. So, let's go. All the questions here are from the prior years. Okay. So, let's concentrate on these and try to answer as much as possible. Okay. So, now coming to the first question, transplantation of tissues or organs. Okay. Tail organs often so this is fails, okay? Fails often due to non-acceptance by the patient's body. Which type of immune response is responsible for such reactions? So our options are cell-mediated immune response, humoral immune response, physiological immune response, and autoimmune response. So let's go. We have a minute to solve this question. Okay. Why do transplantation fail? Oh, this should be easy for me to access things. Transplantation of tissues or organs fails due to non-acceptance by the patient's body. Which type of immune response is responsible for such reactions? All right. So can we go for the answer? So the answer is actually cell-mediated immune response. Okay. So the reason why this particular thing fails whenever the organ donation happens is because of the surface antigens that are present. So whenever a new organ is taken from the donor, so there are certain antigens which are not familiar to our body. So when this comes, our memory cells will be able to differentiate. Our cell-mediated immunity will be able to differentiate between what is self cell and what is non-self cell. So one of the function of our T lymphocyte, okay, our T lymphocyte is to differentiate between self. Okay, sorry, students, I think you might have not heard the voice. So the next question is malt constitutes about what percentage of the lymphoid tissue in human body? And our options are 20%, 70%, 10%, and 50%. Okay. So you have a minute to answer the question. So let's go. MALT stands for mucus associated lymphoid tissue. Okay. All right, students, the time is almost running out. So let's go. All right. It's a very simple question. So let's see if we have the right option. Okay, yeah. So MALT stands for mucus associated lymphoid tissue. So they are very, very important. So they are basically present in your esophageal lining and the lining of your respiratory tract. Okay. So they're very, very responsible for actually protect your body from the food you eat or the incoming air, and they're also present in the genital tracts of you of the human body. So basically their function is to prevent any of the antigens that are coming from the direct entry of food, water, or any of the external source. So they constitute about 50% of the overall lymphoid tissues that are present in our body. Okay, so remember it is 50%. And MALT stands for mucus associated lymphatic tissue. And where they are present? Gastroesophageal tract, and then we have respiratory tract, and then you have urinogenital tract. Understood? So they provide a cellular barrier of immune system, which is non-specific immunity. So let's go for the next question. Okay, so yeah. Which of the following immunoglobulins constitute the largest percentage in human milk? And the options are IgD, IgM, IgA, and IgG. Okay, so which of the following immunoglobulins constitutes the largest percentage in human milk? So you have the following options, and again, you have a minute to answer this question. Let's go, students. Very easy and very easy and very direct question also. Okay. Try to answer this question. Which of the following immunoglobulins? Okay. One more clue I'll give you. Immunoglobulins also stands for antibodies. Okay. All right, students, your time is up. So let's see which is the right option. So the question asked here is which of the following immunoglobulins constitutes the largest percentage in human milk? So basically in the beginning of the lactation period, mother's milk is made up of rich volumes of antibodies, and this is called as colostrum. That's why we emphasize on breastfeeding and how it is very important to build the immunity in the baby. Okay, it's a kind of a passive immunization. So, because direct antibodies are given from mother to the baby to fight against the disease, so the rich amount of antibodies that are present in the mother's milk, that is during the initial stages of lactation, or in the colostrum, that is IgA antibodies. Okay, immunoglobulins A. Okay, next question. If you suspect a major deficiency in antibodies in a person, to which of the following would you look for confirmatory evidence? So if a suspect is having a deficiency of antibodies, okay, which of the confirmatory tests you would perform? Fibrinogen in the plasma, serum albumins, and then we have hemocrites, and then we have serum globulins. Okay, so let's see. If you suspect anybody with antibodies, okay, less antibodies, which of the following tests would you conduct? Again, you have 1 minute, students. Okay, try to answer this question. If where are antibodies found? So that will answer your question. All right, students, our time is up. So let's see which is the right option. So the right option for this question is serum globulins. So checking the serum globulins will give us the exact number of antibodies that are present. So that will show, okay, whenever the person is deficient in antibodies, okay, that means that, okay, he has this problem and that can be tested with the serum globulins. Okay, so this is the test. Next one. Grafted kidney may be rejected in a patient due to, okay, we already answered this question, but again, it's a different type of a question. Grafted kidney may be rejected in a patient due to humoral immune response, cell-mediated immune responses, passive immune responses, and innate immune responses. Okay, so here are the options. So go through it again. So, sorry. So the question is, students, grafted kidney may be rejected in a patient due to humoral immune response, cell-mediated immune response, passive immune response, and innate immune response. So you have a minute to answer this question. So let's go. Why do grafted kidney gets rejected? All right, students. So basically, again, we talk grafted kidney is nothing but a transplantation, right? So the transplantation fails because of what? So the answer is cell-mediated immune responses. As I told you earlier, the cell-mediated immune responses are taken care by T lymphocytes. Okay. So these T lymphocyte cells, they differentiate. Their role is to differentiate what is self and non-self cells. So that will make them to understand, okay, this is not my tissue, so I have to reject it. So that is where the graft failure takes place or the graft rejection takes place. So that is because of the cell-mediated immunity. Understood? So the other options like humoral immune responses, passive immune responses, and innate immune responses are not relevant here. Okay, we know the definitions and meanings of all these things. While coming questions, we'll have questions based on this and see what we can do about these options. Okay. So the next question is, which of the following disease is caused by a protozoan? Very interesting question. So we have the options syphilis, influenza, okay, babesiosis, and then we have blastomycosis. Okay, so we have blastomycosis, and then we have babesiosis, and then influenza, and then syphilis. Okay, so these are the options. So let's go, students. You have a minute to answer this question. So I'm going to pause the video. Please take a minute and then answer the question. It's 30 seconds up, students. We have to hurry. All right. So can we discuss the answers now? Yes. Which of the following disease is caused by protozoan? So let's look at our first option. Syphilis is a bacterial disease. Okay, it's caused by a bacteria. Okay. And then influenza, viral disease. Coming to babesiosis, it is a protozoan disease. So it's usually not so common disease. So what kind of protozoan are we talking about? Protozoan is malaria-like disease, and malaria-like protozoan is causing this particular disease, and it is usually found in the American region, like South America. It is found, and it is not so common. But anyways, the question has been asked, and babesiosis is the answer. Blastomycosis is a fungal disease, and this happens because of the inhalation of spores, okay, which causes an infection. So we know that our answer is babesiosis, which is the option number 3, which is a protozoan disease. Okay, and we have another question, very interesting question again. Which of the following endoparasites of humans show viviparity? So we have our first option, okay, Ancylostoma duodenala, and then we have Enterobius vermicularis, and then we have Trichinella spiralis, and then we have Ascarius lumbricoides. Okay, so these are your options, and this is the question. Which of the following endoparasites show viviparity? Okay, so let's see. Have a minute to answer this question. Let's go, students. Question is, which of the following endoparasites of humans show viviparity? Okay, Ancyclostoma duodenella, Enterobius vermicularis, Trichinella spiralis, and then Ascarius lumbricoides. Okay, so let's go for the answer. So the answer for this question is actually Trichinella spiralis, whereas you have Ancostoma duodenella, and then Enterobus vermicularis and Ascaris lumbricoids. All of them are oviparous and egg-laying parasites, whereas Trichinella spiralis is also called as pigworm. Okay, pigworm. So it comes from eating pig pork. So basically, they lay their young ones directly without laying eggs. So Trichinella spiralis is a parasite that is viviparous without laying eggs. So they basically give birth, and their larvae, okay, infectious larvae is what enters the human body. Understood? So through pork. So that is why the option is option 3 with respect to this question. So let's go for the next question. HIV that causes AIDS first starts destroying B lymphocytes, leukocytes, helper T cells, and then we have thrombocytes. Okay. So look at the question. HIV, human influenza virus. Oh, sorry. Yeah. So that causes AIDS first starts destroying B lymphocytes, leukocytes, and then you have helper T lymphocytes, and then you have thrombocytes. So which is the right option? You just look at it. Okay. You have a minute to answer this question, students. So just try to answer. Okay. Can we go for the answer? So HIV that causes AIDS starts destroying what? So this is a very, very common disease, and we have been studying about HIV in very detail. So we have heard about HIV a lot. So we know that HIV, when it comes initially to our body, it starts destroying a specific type of cells called as helper T lymphocytes. Okay. So helper T lymphocytes, or it is also called as CD4 cells, are very, very important immune cells to establish a contact between cell-mediated immunity and humoral immunity. When this link is broken down, that's when we have acquired immunodeficiency syndrome. So when we don't have the capacity to cause an immune response or protect our body from any of these pathogens, so that is because CD4 cells, or it is also called as helper T lymphocytes, are targeted by the HIV. So which basically turns individual helper T4 cells into a HIV factory. Okay, so that is the answer. Which one? Option 3. So let's go for the next question. The cell-mediated immunity inside the human body is carried out by who? B lymphocytes, thrombocytes, erythrocytes, and T lymphocytes. Very easy. Most of the questions are based on cell-mediated immunity. Okay, so just let's answer this because we already know the answer. We tried to answer 2 of the questions similar to this. So see the question again. Cell-mediated immunity inside the human body is carried out by B lymphocytes, thrombocytes, erythrocytes, and then you have T lymphocytes. Again, students, you have a minute to answer this question. Try to answer. All right, so we know the answer, that is cell-mediated immunity is caused by T lymphocytes, right? So B lymphocytes, they are responsible for humoral immunity, right? Humoral immunity. And then you know that the thrombocytes are, you know, it's like natural defense system without any specificity. And then you have erythrocytes, which are red blood cells. Okay. So thrombocytes are responsible for clotting. Okay, clotting mechanism. So B lymphocytes, T lymphocytes is important for cell-mediated immunity. Okay, cell-mediated immunity. All right, students. So can we go for the next question? Identify the site where Ucheria bancrofti is normally found on human body. Muscles of the legs, blood vessels of the thigh region, skin between the fingers, and the lymphatic vessels of the lower limbs. Identify the site where Ucheria bancrofti, okay, is normally found in human body. So you have your options. Is it the muscles of the legs? Is it the blood vessels of the thigh region? Is it the skin between your fingers or the lymphatic vessels of your lower limbs? Again, you have a minute to answer this question. Take your time, read the question properly, and try to answer as soon as possible. So these kind of questions are quite direct, students. Okay, so it's quite direct questions. So most of the questions that is coming from your human health and disease is quite direct. Apart from few things that we saw, like which is a OB parasite is one type of a question. So what kind of examination when you do when you have low antibodies in the human body? So these kind of questions are quite interlinked questions. It's okay. The more we solve, the more we get okay the information. But try to answer this question. Identify the site where Cherry Bancroft is normally found in human body. Okay. All right. So can we answer this question? So the answer is lymphatic vessels of the lower limbs. So I have already told you what is lymph nodes and where the lymphatic vessels are present. And what happens when the Wucheria bancrofti, which is a nematode that enters to our body through through a mosquito, right? So it enters through a mosquito and then it targets our lymphatic vessels and lymph nodes, especially of the lower limbs. That leads to chronic inflammation of the lower limbs as well as the one of the genital organs like testis. So which leads to deformity in the parts of our lower limbs. So our answer is lymphatic vessels of our lower limbs. And skin between the fingers, it is for ringworm. Okay, this is for ringworm, usually most commonly. Blood vessels of the thigh regions and muscles of the legs, no. We don't have a specific target for that. So we'll neglect it. So our next question is, which one of the following is a hallucinogenic drug? So our options are caffeine, morphine, lysergic acid diethylamide, and then we have opium. Okay, which one of the following is a hallucinogenic drug? Caffeine, morphine, lysergic acid diethylamide, and then we have opium. Okay, so go through it again, students. You have a minute to answer this question. Try to do it as soon as possible. In our textbook, it's a small part of it is given. So we should be able to remember this. Okay, so let's see which one of the following is a hallucinogenic drug: caffeine, morphine, lysergic acid diethylamide, and opium. So lysergic acid diethylamide, which is also called as LSD, is a hallucinogenic drug. Okay, so which causes visual impairment, and then a person goes into euphoria and he loses the sense of reality. Okay, so that is basically extracted from a ergot fungi. Okay, so it's taken orally. Anyways, so that causes hallucinogenic experiences in the individual that take this. Whereas caffeine increases adrenaline rush, morphine is a sedative, opium is a painkiller. Okay, so we know what the other option. So correct option is option 3. Along with the hallucinogenic comes like mushrooms, and then you have datura, which is another hallucinogenic category of drugs that are used. Okay, next one. Yeah. Which one of the following is not the property of a cancerous cells, whereas the remaining three are? They compete with the normal cells for vital nutrients. They do not remain confined to a particular region in the formation, in the area of formation. They divide in an uncontrolled manner, and they show contact inhibition. So we have to find which is not the property of cancerous cells. So we have four options here. They compete with normal cells for vital nutrients. They do not remain confined in the area of formation. They divide in an uncontrolled manner. They show contact inhibition. Okay. So here are the options, students. So let's go and answer which is not the property of cancerous cells. Okay. Did you get the answer right? All right. So we are talking about cancerous cells. So we know what are cancerous cells, right? Any cells which has lost its capacity to stop dividing. So now that will lead to cancer. So usually in our body there is a proper number or proper volumes or proper area to which the cells have to divide. So and the division rate and the way they stop the division happens because of something called as contact inhibition. So the moment they come in contact, okay, and they're packed, so that will create an inhibition process where the cells will stop dividing. Understood. And many of the tissues in our body have permanent, they do not regenerate. And once they're damaged, they're gone. But once they turn cancerous, they keep on producing into this mass of cells, which are quite vigorous in extracting the nutrients from the surrounding cells. And they have their own, they develop their blood vessels, and where the nutrients are, sucked away from the region. Okay, and that is when they starve the rest of the cells. So basically, they first option, that is they compete with normal cells for vital nutrients. Yes, that is correct, and they do not remain in confined area of formation. Yes, there is a type of cancer which is called as malignancy, where it causes metastasis. That is, the cells from where they have originated, they detach and they migrate to the other parts, and then they cause cancer in that different region. That is called as malignancy, and they participate in malignancy. They divide in an uncontrolled manner. Yes, they don't have contact inhibition, and they divide in a very huge mass. So that is another point. And they show contact inhibition. No, normal cells show contact inhibition. Cancer cells do not have contact inhibition. Contact inhibition is something that, as I told you, once the cells have reached the specific numbers, once they are in contact with the rest of the cells, they can feel the neighbor cells and they inhibit each other's cell division. So this is called as contact inhibition, and the cancer cells do not have contact inhibition. So our option is option 4. So let's go for next question. Identify the wrong statement with reference to immunity. Okay, so we have the first statement: active immunity is quick and gives full response. Fetus receives some antibodies from mother. It is an example for passive immunity. When exposed to antigen, living or dead, antibodies are produced in the host body. It is called as active immunity. When ready-made antibodies are directly given, it is called as passive immunity. Very important statements. Okay, so that shows how much of understanding we have about immunology. So look at this question, students, and try to answer this. So let's go for once again and read this question. Identify the wrong statement with respect to immunity. Active immunity is quick and gives full response. Fetus receives some antibodies from mother. It is an example for passive immunity. When exposed to antigen, living or dead, antibodies are produced in the host body. It is called as active immunity. When ready-made antibodies are directly given, it is called as passive immunity. So Again, you have a minute to answer this question. So try to look at this question and please answer. All right, students, can we try to answer the question? Let's go from the bottom for a change. When a ready-made antibodies are given, directly given, it is called as passive immunity. Isn't it so? So whenever, for example, we have passive immunization for tetanus, anti-tetanus, injection. So tetanus is an example for passive immunity. Anti-venom, when a snake bite has happened, okay, when the snake has bitten you, so you give directly anti-venom. Preformed antibodies are given. So that is another example. Then again, another example is mother's milk, right? Mother's milk is another example for this. Okay. Next one. So that is called as passive immunity. This is right. Absolutely right. When exposed to an antigen, living or dead, antibodies are formed in the host body. It is called as active immunity. Yes, it is a very gradual process. That is when we are given examples like polio vaccine, DTP vaccine, BCG vaccine. So all this vaccination or immunization is based on deliberately introducing living or dead, microorganisms that cause a disease that will generally develop a memory of this particular organism. And when the real organism comes, which can cause an infection, that will be killed. Okay. So that process of developing a memory and then targeting these microorganisms where our body only will produce these antibodies, that is called as active immunization or active immunity. So that is C is also correct. Fetus receives some antibodies from mother. It is an example for passive immunity, as I mentioned here. This is also right. Coming to the last option, okay, coming to this option, active immunity is quick and gives you full response. So, is it quick? No. It gives full response. True. It's not quick. It takes years or months or days, okay, for the body to build memory, to develop antibodies, okay, and to target and finally get immunized. Okay, so it is a very slow, very slow process. It takes a lot of time. So that is why option A is a correct option when it comes to the wrong statement with respect to immunity. Okay, so that was answered in 2020. Next one. The infectious stage of plasmodium that enters the human body is, this should be an easy question. So the options are female gametocytes, male gametocytes, trypanocytes, and then we have sporozoites. Okay. So here are your options, students. So you have a minute to answer this question. Please go through the question again. The infectious stage of plasmodium that enters the human body is female gametocyte, male gametocyte, trypanocytes, and then we have sporozoites. Okay. We have discussed this earlier, so it shouldn't be that difficult. All right. Can we go for the answer? What is the infectious stage of plasmodium? Simple, sporozoites. So the mosquito bites a healthy person. From the salivary glands of the mosquito comes the infectious stage of this plasmodium's life cycle, that is sporozoites. The sporozoites migrate from the salivary glands all the way to the blood vessels. From blood vessels, it will go to the liver, right? So that is when the infection starts. Okay. So that is sporozoites is the correct answer. So what happens after it enters? It goes to, see, sporozoites goes to liver, and it will, what it will do, it will incubate, okay, or it is called as hepatic hibernation or hepatic incubation. So after this, what will happen? Now they will start dividing asexually in the liver. From liver, they will go and migrate to the, they will migrate to the red blood cells. And in red blood cells, when these, okay, when these cells, they attach to the red blood cells, this is called as a trypanocyte. In trypanocyte, they will divide, okay, asexually and form male gametocyte and female gametocyte. So you understood what are the different things, and our correct option for this question is option D, that is sporozoites. Okay, so let's go for the next question. Identify the correct pair representing the causative agent of typhoid fever and the confirmatory test for the typhoid. First option is Plasmodium vivax and UTI test, Salmonella typhi, that is Widal test, Streptococcus pneumonia, Widal test, Salmonella typhi, and then we have Anthrone test. Okay, so here are our option. And our question is, we have to find out what is the causative agent of typhoid and the test for typhoid. Okay, so 2019 question. So let's go, students. It's very, very easy. And it is our first disease that we studied also, right? So try to answer this. All right, can we see which is the right option? So typhoid is caused by Salmonella typhi. And the test for typhoid is Widal test. What is Widal test? I also told you this one. Triple antigen test is also called as Widal test. So that is what is a test is is used to detect the presence of your Salmonella typhi. Understood? So that is a causative agent, and that is the test that is for the particular disease. Next one. Antivenom contains preformed antibodies, while polio drops that are administered into the body contain what? Activated pathogens, harvested antibodies, gamma globulins, attenuated pathogens. Okay. So question says, when you talk about antivenoms, it contains preformed antibodies. What do polio vaccines contain? Okay, so here are our options. Take a look at it and try to answer this question. All right, students. What does the vaccines contain? Any vaccine contain? Vaccines contain attenuated pathogen. What is attenuation? Making the pathogen inactive of, you just disalarming this pathogen. They do not have the capacity to cause a disease, but the same pathogen which causes a disease, a part of a pathogen, okay, is used for, introducing into your body so that your body learns what kind of an antigen it is. For example, in polio, live attenuated pathogen is used. That means what? Polio virus, which is alive, attenuated means what? It has lost the capacity of an infection. That is used for the vaccine purpose. Okay, so in polio vaccine, attenuated pathogens are used, not the rest of it. Okay, next one. In higher vertebrates, the immune system can distinguish between cells and non-cell cells. It is if this property is lost due to genetic abnormality and it attacks cells, then it leads to what? Okay, allergic response, graft rejection, autoimmune disease, and then we have active immunity. So let's go, students. What happens if in higher vertebrates, okay, an immune system loses its ability to distinguish between cells and non-cell cells? Okay, so if the property is lost, what it will lead to? What does it lead to? Do we have answers? Okay. So basically, when higher vertebrates, we have a capacity to differentiate between cells and non-cell cells. So if you talk about human beings, we have it through cell-mediated immunity, or T lymphocytes do the job. Whenever this property is lost, it leads to a disease called as autoimmune diseases. So autoimmune diseases can be rheumatoid arthritis, psoriasis, okay? So Alzheimer's disease, So here what will happen, our own tissues like brain cells, the bone tissue, or the epidermal layer of your skin, any of the tissue pertaining to a specific disease, so or these parts are targeted by your own cells, thinking that it is an antigen, thinking that it is an external cell, tissue. So when that happens, our own self destruction of the tissues will take place with respect to that particular body which is affected. So that disease is called as autoimmune disorder. Example is rheumatoid arthritis, Alzheimer's disease, okay, and then psoriasis is another example. So this is about this question. Okay, next one. The active form of entamoeba histolytica feeds upon, very important question, mucus and submucus of colon only, food in the intestine, blood only, erythrocytes, mucosa and submucosa of colon. Okay, so what does an active form of entamoeba histolytica feeds upon? So we have four options. What is the food for your entamoeba histolytica? Okay. All right, can we see the answer? When we talk about entamoeba histolytica causes what? Amoeboic dysentery or amoeboid dysentery. So basically, it enters through housefly. Housefly is the vector. Okay, so basically, it sits on the fecal matter and then it sits on food or water. Then we ingest it. So the amoebospores enter in our large intestine and it bursts open and large amount of amoeba is released. Here, what happened? The amoeba will have to survive. So they basically attack your red blood cells, mucosa, and submucosa of the colon. So I think you should know what is mucosa, submucosa of the colon. If you take the cross section of your large intestine, what do you get? You get submucosa, mucosa, muscularis, and serosa or adventitia. Okay, if you remember, the cross section of your intestines or alimentary canal shows what? First one is mucosa, right? And the second one is submucosa. And third one is muscularis. And then you have serosa. I hope you can see this. Okay. So they target these two layers, plus they also target your red blood cells. Okay, sorry. They also target your red blood cells or erythrocytes. All right. So that is about entamoeba histolytica. So, okay. The final question. Asthma may be attributed to bacterial infection in the lungs, allergic reactions of the mast cells in the lungs, inflammation of the trachea, accumulation of fluid in the lungs. Okay, very easy question. It will be tricky, but you can answer. Asthma is attributed to, as you can get asthma because of what? Bacterial infection in the lungs, allergic reaction in the mast cells in the lungs, inflammation of the trachea, and accumulation of fluid in the lungs. Okay, so let's go, students, answer this question. Okay. All right, let's look into it. When we talk about asthma, asthma is also called as what? Over exaggerated immune response, we'll call it as. What is over exaggeration? So, our body, whenever an antigen enters, it can be dust, it can be pollen, it can be any of the allergies you have, like, for example, peanut allergy. Okay, you have peanut allergy, you have lactose intolerance, or lactose allergy, or allergies to medication. Okay, so what happens? Whenever any of these factors enter our body overthinks that this is a very big problem, and a lot of antibodies are released, and those antibodies are called as IgE. And basically, when the IgE is released, it will create a lot of nuisance in our body, such as rashes, irritation in the eye, running nose, burning sensation. Okay, so many different types of, for example, breathing difficulties in case of asthma. When it comes to asthma, which is an allergic reaction and which is because of several reasons, some people get asthma because of dust, Some people get asthma because of the congestion, because of the pollen grains, etc. What happens whenever these allergens are detected in our lungs, the mast cells will release a lot of this IgE. So that is the answer. Allergic reaction of the mast cells will result in asthmatic problems in an individual suffering from asthma. Okay, so our option is B. Bacterial infection of the lung is again with respect to pneumonia. Inflammation of the trachea is not here. Okay. Anyways, accumulation of the fluid in the lungs is again pneumonia, so it's not asthma. So, anyways, we come to the end of our question and answer session with respect to human health and disease. I recommend you students to solve as many questions as possible with respect to this chapter. It's kind of an important chapter, especially when it comes to immunity and the type of drugs, okay, and the names of the diseases and the pathogens that cause them. It's very easy. It's a very scoring, section when it comes to NEET examination. Please don't take it too casual, but when it's easy, you can just practice it in your free time, okay, and get the things right. All right. So anyway, students, I'll meet you tomorrow in tomorrow's class. Thank you very much. If you have any doubts, you can leave. I'll be here for a few minutes.